Blood facts, functions and composition. Blood is a specialized bodily fluid in animals that delivers necessary substances such as nutrients and oxygen to the cells and transports metabolic waste products away from those same cells. Blood facts, functions of blood composition of blood plasmaProteinsAmino acids nitrogenous wasteNutrientsGasesElectrolytesRed blood cellsWhite blood cells granulocyte granulocytes platelets vascular spasmPlatelet plug formationCoagulationProduction of blood hemopoiesis erythropoiesis thrombo. Poiesisaging changes in the blood. Blood facts. Approximately 8% of an adult's body weight is made up of blood. Females have around 4-5 liters, while males have around 5-6 liters. This difference is mainly due to the differences in body size between men and women. Its mean temperature is 38 degrees Celsius. It has a pH of 7.35–7.45, making it slightly basic. Less than 7 is considered acidic. Whole blood is about 4.5–5.5 times as viscous as water, indicating that it is more resistant to flow than water. This viscosity is vital to the function of blood because if blood flows too easily or with too much resistance, it can strain the heart and lead to severe cardiovascular problems. Blood in the arteries is a brighter red than blood in the veins because of the higher levels of oxygen found in the arteries. An artificial substitute for human blood has not been found. Functions of blood. Blood has three main functions: transport, protection and regulation. Transport. Blood transports the following substances: gases, namely oxygen, O2, and carbon dioxide, CO2, between the lungs and rest of the body, nutrients from the digestive tract and storage sites to the rest of the body, waste products to be detoxified or removed by the liver and kidney, hormones from the glands in which they are produced to their target cell sheet to the skin so as to help regulate body temperature. Protection. Blood has several roles in inflammation. Leukocytes, or white blood cells, destroy invading microorganisms and cancer cells. Antibodies and other proteins destroy pathogenic substances. Platelet factors initiate blood clotting and help minimize blood loss. Regulation. Blood helps regulate pH by interacting with acids and base water balance by transferring water to and from tissues. Composition of blood. Blood is classified as a connective tissue and consists of two main components. Plasma, which is a clear extracellular fluid-formed elements, which are made up of the blood cells and platelets. The formed elements are so named because they are enclosed in a plasma membrane and have a definite structure and shape. All formed elements are cells except for the platelets, which tiny fragments of bone marrow cells. Formed elements are erythrocytes, also known as red blood cells, RBCs, leukocytes, also known as white blood cells, WBCs, platelets. Leukocytes are further classified into two subcategories called granulocytes, which consist of neutrophils, eosinophils and basophils, and agranulocytes, which consist of lymphocytes and monocytes. The formed elements can be separated from plasma by centrifuge, where a blood sample is spun for a few minutes in a tube to separate its components according to their densities. RBCs are denser than plasma, and so become packed into the bottom of the tube to make up 45% of total volume. This volume is known as the hematocrit. WBCs and platelets form a narrow cream-colored coat known as the buffy coat immediately above the RBCs. Finally, the plasma makes up the top of the tube, which is a pale yellow color and contains just under 55% of the total volume. Blood plasma. Blood plasma is a mixture of proteins, enzymes, nutrients, wastes, hormones and gases. The specific composition and function of its components are as follows. Proteins. These are the most abundant substance in plasma by weight and play a part in a variety of roles including clotting, defense and transport. Collectively, they serve several functions. They are an important reserve supply of amino acids for cell nutrition. Cells called macrophages in the liver, gut, spleen, lungs and lymphatic tissue can break down plasma proteins so as to release their amino acids. These amino acids are used by other cells to synthesize new products. Plasma proteins also serve as carriers for other molecules. Many types of small molecules bind to specific plasma proteins and are transported from the organs that absorb these proteins to other tissues for utilization. The proteins also help to keep the blood slightly basic at a stable pH. They do this by functioning as weak bases themselves to bind excess H+ ions. By doing so, they remove excess H+ from the blood, which keeps it slightly basic. The plasma proteins interact in specific ways to cause the blood to coagulate, which is part of the body's response to injury to the blood vessels, also known as vascular injury, and helps protect against the loss of blood and invasion by foreign microorganisms and viruses. Plasma proteins govern the distribution of water between the blood and tissue fluid by producing what is known as a colloid osmotic pressure. There are three major categories of plasma proteins, and each individual type of proteins has its own specific properties and functions in addition to their overall collective role. Albumins, which are the smallest and most abundant plasma proteins. Reductions in plasma albumin content can result in a loss of fluid from the blood and a gain of fluid in the interstitial space within the tissue, which may occur in nutritional, liver and kidney disease. Albumin also helps many substances dissolve in the plasma by binding to them, hence playing an important role in plasma transport of substances such as drugs, hormones and fatty acids. Globulins, which can be subdivided into three classes from smallest to largest in molecular weight into alpha, beta and gamma globulins. The globulins include high-density lipoproteins, HDL, an alpha-1 globulin, and low-density lipoproteins, LDL, a beta-1 globulin. HDL functions in lipid transport, carrying fats to cells for use in energy metabolism, membrane reconstruction and hormone function. HDLs also appear to prevent cholesterol from invading and settling in the walls of arteries. LDL carries cholesterol and fats to tissues for use in manufacturing steroid hormones and building cell membranes, but it also favors the deposition of cholesterol in arterial walls and thus appears to play a role in disease of the blood vessels and heart. HDL and LDL therefore play important parts in the regulation of cholesterol and hence have a large impact on cardiovascular disease. Fibrinogen, which is a soluble precursor of a sticky protein called fibrin, which forms the framework of blood clot. Fibrin plays a key role in coagulation of blood, which is discussed later in this article under platelets.